Okay, hello, welcome back. So we'll move on with the lecture 2. I'll move to the lecture mode. All right, okay. So, well, here are my details again. Okay, moving on to this. What I want to talk to you about. Okay, this is what I mentioned briefly in the first video about how the structure of the program. So intro lectures week 1 and week 2. So now I think because I missed week 1 and 2, so we'll probably go into week 3. Then you'll be required to identify a topic of interest, and then this will, I think, will be in the form of spectrum assignment 1, where you have to key in your little bit of explanation of what your topic is about and what are your sources, your informal sources. It doesn't matter, maybe your source is probably your conversation with a friend, so it doesn't really matter that much. And then which I will give a response to, right? Assessment and response of assignment 1. And then you will move on to setting up the title of your mini research that you're going to do, or if the title that you identified in the assignment is not suitable, I may ask you to re-identify a new topic of interest. And then the following weeks will be, once you set the title, it will be looking for articles and of course reading it, and then pertaining to your mini topic. This will probably take you to 2 to 3 weeks, in which we usually don't have a class. If there is a class, remember I mentioned that okay, there will be class sessions where I can be there, and it's up to you to come. Maybe if you want to discuss with me anything about your title, so it's not really a problem. So then we come to your spectrum assignment number 2, in which you will be asked to describe 3 papers. Well, for now it's 3 papers. You have to upload the papers, tell them what the paper is about, answer some questions, and I think this will be done somewhere in week 7 or week 8. Again, a week after that, if I'm efficient enough, I'll probably give you a response. If I don't give you a response on a spectrum itself, I'll probably give you a response by WhatsApp or whatever means available, and that will take us up to week 9. And then for which I will also talk to you about how to prepare for your 3-minute presentation and oral assessment, and that will take place sometime in week 10 or week 11. The first time I did that, we had 2 examiners, independent examiners, so that means you have to present with two different people. Last year, the last semester I didn't have time to organize that event, so I did most of the individual assessment, but everybody has to go through that assignment, oral assessment process. And then the following weeks, of course, for you to prepare your written assignment, in which the actual oral assignment will take place between week 12 or week 13, and then following on from that, you should be ready with your full assignment. Remember, it's a Turnitin assignment, so that means you will have to submit after the Turnitin report. I will only allow what was it last year? Maybe about 25%, 20 to 20%. What was it? 22% matches. And if you submit with higher matches, then you get more marks deducted. Alright, so basically that's the whole running of your program. Okay, if you have any questions, please do not hesitate and ask me through the WhatsApp group. But if you're not comfortable, you can send your messages individually. Okay, alright, so moving on. Right, okay, obviously this course talks about the DNA and you, the DNA technology. No, I'm not asking you to remember what these things are, but essentially these are molecules of DNA, and they are in your cells. Alright, okay, let's look at the cell. So for a human being, right? We have roughly about 40 trillion cells, right? Million, 1 million million, right? Okay, 1000 million is 1 billion, so this is 40 trillion cells. I hope I'm right. Okay, but if I'm not, it's okay, don't worry. So it's like 40 trillion cells. Each cell will have your DNA. Of course, there are exceptions, but we're not bothered about that. So assuming that each cell have your DNA, so this is your cell. Normally your cells will have a nucleus, and within the nucleus is where you get your chromosomes, right? Each individual cell will have, a human cell will have 46 chromosomes, and whether you have XY, if you have XY you are a male, if you have XX you're a female. The rest are chromosomes that determine different aspects of your the features that you carry. So this chromosome, each of the chromosome is actually made up of DNA that has been wound on histones. Histones are kinds of proteins, and wound among themselves, and then wound again, you know, multiple levels of winding until you get a chromosome. And if you unwind that, then you'll get the double-stranded DNA, which is a double-stranded helix. I mean, this is a very popular picture or diagram that depicts your inheritance. And basically there are double-stranded, and this double-stranded DNA is like a zip, you know, a zipper. They attracted to each other, and this attraction is based on some compounds. The four main compounds are GATC: guanine, adenine, cytosine, and thymine. They have a attraction. A G is always attracted to a C, and A is always attracted to a T, and this gives you the basis of what the DNA is all about. All right. So essentially, because of its it has the DNA, it has the ATGCs and on. Essentially, a DNA molecule contains information. Let's imagine that each chromosome is a book. Okay, and because each chromosome is a book, that means individually inside your cells you have about 46 books. All right. The 46 chromosomes are actually made up of pairs. So you have a pair of chromosome number 1, a pair of chromosome number 2, a pair of chromosome number 3, and so on. With the last pair, this is what you call as your sex chromosomes. So you have pair 1, 2, 3 until 22, and then you have your sex chromosome. In this case, they are both X's, so you can guess that this is actually a female individual. If it is an X and Y, so there's a smaller chromosome, one big and one small, but they're still called a pair. And because of that, so that means you have a duplicate of each book in every cell. So the reading or the understanding of the human genome, a genome is the whole content of the human DNA. So the genome is really about the Human Genome Project is really about trying to understand the book, right? And there are in all kinds of books, even in literature and so on, or if it's in a different language. First of all, you need to learn to read it, and then after reading a bit with the ability to read it, then you can try to understand it. All right. So there are two stages to it. So one of the first objectives of the Human Genome Project is to actually read the entire human book of life. All right. So that started the Human Genome Project. Started in about 1980s, and then it the human genome, it actually, the first stage of completion was done in 2003, and then eventually the chromosomes were all the genome is fully sequenced, I think, if I'm not mistaken, it's 2022. All right, that you actually have all the things that is being read. Okay, the reason why there are stages of completion, you know, in 2003 they say it's completion stage 1, because it very much depends on the technology of sequencing that was available then. All right, I will talk about that in a bit. So anyway, sequencing is a process of reading the genomes, reading the sequence. So the Human Genome Project is about reading the genome, the human genome sequence, but the project is also carried out to read the genomes of other organisms as well, like I'll show you in a bit. So after reading the human genome, they found out that each, if you just take one chromosome, one yeah, one chromosome of each pair, right, and then you string all the letters together, all the letters will make up about 3,000 million letters, which is about 3 billion. Okay, 3 billion base pairs for a haploid genome. Haploid means a single. Um, it's not. When it comes in pairs, it's called a diploid. But if you just take one of each, then it's called a haploid. So 3 billion base pairs of sequence is what you can see in the human cell. For comparison, we have 3 billion. So the yeast, you know, the Saccharomyces cerevisiae is the yeast, the type of yeast that you use in making bread. Yeah, is a yeast. So that organism contains about 12 million. Drosophila melanogaster, this is the group of flies that attack fruits. You know, if you leave your bananas on the table, for where if it gets a bit too ripe, and then you'll see all these tiny flies that goes to it. So that's Drosophila. That one has got 130 million. This is the housefly. I've got a question mark there for you to guess. This is the bacteria E. coli, that causes a lot of cholera cases and so on. So it's it's 4.6 million, and so on and so on and so forth. So if you like to guess what will be the sizes of the housefly, what would be the sizes of the house rat or mouse, and then what are the size of the onions? The housefly is about 900. The rat, Mus musculus, the mouse, is about 2,700, which is not so far from us. But if you look at this onion, this very humble onion has a genome size of about 17,000 million. It's about what, 5 times more than ours? Okay, so does that mean that they have more DNA? That means the average onion is more intelligent than the human being? Well, not really, because it very much depends on how the genome is being used, right? So let's not worry about that. So but this slide is just for you to give an idea of the amount of genetic material that is present in all these different organisms. So reading the genome is one thing, understanding it is another story. Okay, so this is a book. Just trying to imagine how a book looks like. I'm sure you've all seen books. You know, it's got every letter, every sentence can be seen clearly. It starts with a capital letter. You have chapters, you have subsections, you have page number, and you have the contents. This is a book, but it's very much different from the book of life, your DNA, in which your DNA actually looks very much just like this, right? It's pages and pages and pages of just letters, right? it doesn't even have any information about where to start reading, where does one chapter start, where does one chapter end, and so on. So it doesn't have that. So that's why reading the sequence is one phase, and then understanding it is another. So imagining the genome, right? You have 3 billion base pairs. To imagine that, I think, you know, if you type it out on a piece of paper, double spacing, font size 12, you actually need about 15 reams of paper to actually print that out. 3 billion letters, 15 reams, you know, one ream is that thick, and then you have 15 reams to get that. So so obviously what the thing that you need to do with that is to actually look at, try and read, and then start to decide, okay, this chapter, this portion, right? For example, here this is about one chapter, this is another chapter, and so on. So that's how you do it, and then you go on and then decide. And what's worse, I don't know whether you're aware of this, some chapters, some chapters inside the human book of life is organized in subsections. So that means one story is like, you know, it's on this page, like 3 sections here, and then it joins up with another section somewhere else. Yeah, that's how the genome is actually being organized. So they have to figure all that out. And then of course when I talk to you here, I say, you know, chapters and subsections, but in genetic terms, this is what we call as genes. I'm sure you've heard of it, you know, the gene that determines your eye color, what gene determines your skin color, your hair color, the gene that causes you know, cancer and things like that. There are various types of genes. So basically understanding the human genome means trying to figure out all these genes and how they work. So the purpose, the end target of a human genome project is, again, if it's like a book, you want to be able to mark the book and say, okay, this chapter is here, this chapter is. So that's what you want to do, assuming that, for example, here you can see this is the chromosome X. You want to be able to map, or at the top there is a gene for ichthyosis, there's a gene for Duchenne muscular dystrophy, there's a gene here that causes retinitis pigmentosa. Retinitis pigmentosa is a kind of eye disease. I don't know whether you've heard of Fragile X syndrome, you know. So basically you're trying to map the position and the function of the gene. So that's the whole idea of the Human Genome Project. And overall, it has been they counted, and then they said that, you know, overall for humans, in the 3 billion base pairs of DNA that you can see in humans, it contains about 21 to about 25,000 genes that makes the human being. Okay. I don't know whether I want to go into this, but how genes work, right? A little bit of biology here. How genes work is that, as I mentioned, every cell will have your DNA, right? But in each of the cells, for example, in your retina, you will be reading the portion that is involved for the eye function. If it's for the skin, then for the skin function it will read maybe a different a different section of the genome. I mean, it's like a library, right? It contains all information. If a science student goes to the library, you know, I will go into the science section. But if maybe some of you from law or from religious studies, you know, you go to your own section, and then you just read what you want based on what you need. So it's the same thing with your cells. If you, each and every part of your cells will have all the DNA, but for function, it only reads what it needs to read. Okay, and how it reads that is it opens up the book, the DNA, makes a copy of it, and then that copy is then transported to the cell and used. So your DNA remains intact, right? So you don't take the DNA out and so on. Excuse me. I don't know whether when I was studying when we use the library, there's one place that's called the red spot, in the library, where for books in the red spot, because it's very hot. It's not that the book is hot, but there's a lot of requests for the book, so you cannot take it out, but you can take it to make a photocopy, and then you'll have to return it within an hour or so. So you take it out, and then you copy what you need, and then you return the book so that other people can use it. So that's very much how the DNA functions. Okay. Yeah, I won't also go into this. This is, remember I mentioned that sometimes the gene itself is organized the gene is organized in sections. So sometimes, you know, section 1, section 2, section 3, section 4. When you make a copy, then the copy will be modified to remove the irrelevant sections. So this is just a functioning of how the gene. I don't think I'm going to go into this also, but basically, for your knowledge, this is how a gene is being used. This is your, if you remember the earlier diagram where I showed you the cell. This is the nucleus. The genes are inside the nucleus. If you want to read, so you have an enzyme that makes a copy. This is a copy messenger RNA, and this messenger RNA will be transported out and then used to make proteins. The DNA remains inside. All right. Okay. This is the process. This is the mRNA, which is copied from the DNA, and then this process is called translation. Just to go back a bit, this process here, reading of the DNA to mRNA, is called transcription, and then from RNA to make protein is called translation. Right. And this is the genetic code. The DNA, remember, if you remember what I mentioned, the DNA is made up of 4 bases: adenine, guanine, cytosine, and thymine. Only 4. Yes, the book of life only uses 4 alphabets. For comparison purposes, our books in our language, how many alphabets do we use? We use 26. There are 26 alphabets in our language, right? From A, B, C, D, E, F, G. But in the DNA, in the genetics or textbooks of life, you only use 4 bases. But these 4 bases are arranged in a code, which is called the genetic code, and then you can read it from here, meaning if it's UUU, it's called phenylalanine. If UUC, it also means phenylalanine, but UUA means leucine. So this is what you call as the genetic code wheel or whatever. But this is how you read it for, again. So it's G is glycine. GGU is also glycine, but GUA is valine. So these are all amino acids. All right. If you go back one slide, mRNA, the language of A, T, G's, and C's, will be translated into amino acids. This is where you get the alanine, arginine, asparagine, and this now forms your proteins, the protein that makes you, all right, your enzymes and so on. Okay.